Hello biology students, this is chapter 9, cellular respiration. In section 9.1 we are going to examine the chemical pathways of cellular respiration. So what do we see in section 9.1? We know that food serves as a source of raw materials for the cells in our body and a source of energy. So here we could see a little chipmunk there, or a squirrel eating a plant. So we have an animal and plant. Here's what that plant cell will look like. Here's an animal cell, typical. And in both of those, whether you're a heterotroph like the animal or an autotroph like the plant, both of those cells contain mitochondria. And a lot of what we're going to talk about in this chapter does take place within the cells, and in particular the organelle, the mitochondria. So both plants and animal cells carry out the final stages of cellular respiration in the mitochondria. What's happening inside the mitochondria itself? If you'd like to draw this picture, do so in your notebook. It's important to know the structure of the mitochondria. But looking at the structure itself, the mitochondria does have an outer membrane, and then we have an inner membrane here, and those inner membrane is folded. Those are called the cristae of the mitochondria, C-R-I-S-T-A-E, and those inner foldings are going to help increase the surface area for the reaction to take place. The area between the outer membrane and the inner membrane is called the intermembrane space. And then if you come inside, the area on the inside of that inner membrane is known as the matrix of the mitochondria. So it is important to know the mitochondrial structure because the end reactions of cellular respiration are going to take place in various areas of the mitochondria itself. Remember, when we were doing cellular structure and function, the mitochondria is considered the powerhouse of the cell because it produces ATP via this process. So, chemical energy in food. We know that 1 gram of sugar, or glucose, C6H12O6, when burned in the presence of oxygen is going to release 3,811 calories of heat. We've learned from lab that a calorie is the amount of energy needed to raise the temperature of 1 gram of water by 1 degree Celsius. So let's apply that concept to cellular respiration. Ultimately, here's an overview of cell respiration. If we are in the cell, glucose is coming in, and once it enters the cell, we'd say that glucose is now in the cytoplasm of the cell. And glucose is going to be initially broken down via a process called glycolysis. Glycolysis, the end product there is going to be pyruvic acid. And if oxygen is available, pyruvic acid is going to enter into the mitochondria, where you're going to have both the Krebs cycle and the electron transport chain take place. You have heard of the electron transport chain before. You also heard of electron carriers from photosynthesis, except here we're introduced to 2 new electron carrier molecules. We are going to be introduced to both NADH and FADH2. So ultimately, what do we get then out of this entire process is ATP. And that's the whole purpose of cellular respiration, to produce those ATP molecules. So cellular respiration is the process that is releasing energy by breaking down glucose and other food molecules in the presence of oxygen. This is a key concept. The equation for cellular respiration is 6O2, oxygen, plus C6H12O6, which is glucose, is going to yield 6CO2 plus 6H2O plus energy. This equation, if you recognize it, is the reverse of photosynthesis. So the first step in this process is a process called glycolysis. Glycolysis takes place in the cytoplasm of the cell. the Krebs cycle and the electron transport chain take place in the mitochondria. What happens during the process of glycolysis? Key concept. ATP production at the beginning of glycolysis, the cell is going to use up 2 molecules of ATP to start the reaction. That is going to lead to another molecule in there that's an intermediate, and then eventually that molecule is going to split. So in the first phase of glycolysis, you're going to use 2 ATP to get the reaction started. Then, in the second phase, you're going to gain energy, and at the end of that, you get 2 pyruvic acid molecules, one here and one here. So you get a total of 4 ATP out. So when glycolysis is complete, 4 ATP molecules have been produced. However, because you initially put in 2 ATP molecules here, and you're getting 4 out here, this only gives the cell a net gain of 2 ATP molecules. You also produce a molecule called NADH. One reaction of glycolysis removes 4 high-energy electrons, passing them to an electron carrier molecule called NAD+. Each NAD+ is going to accept the pair of high-energy electrons to become a molecule of NADH. The NADH molecules holds the electrons until they can be transferred to other molecules. Ultimately, that we're going to see in the electron transport chain. So what are the advantages of glycolysis? The process of glycolysis is so fast that cells can produce thousands of ATP molecules in a few milliseconds. Glycolysis does not require oxygen. At the end of glycolysis, pyruvate is formed. The fate of pyruvate at the end of glycolysis, so what happens after glycolysis, depends on whether oxygen gas is available or not. If oxygen gas is not available, we have a process taking place called fermentation. So when oxygen is not present, glycolysis is followed by a different pathway. The combined process of this pathway and glycolysis is called fermentation. Fermentation releases energy from food molecules by producing ATP in the absence of oxygen. This type of respiration is called anaerobic respiration. During fermentation, cells convert NADH to NAD+ by passing high-energy electrons back to pyruvic acid. This action is going to convert NADH back into NAD+ and allows glycolysis to continue producing a steady supply of ATP. Fermentation does not require oxygen. It is an anaerobic process. There are 2 main types of fermentation. The first type is called alcoholic fermentation. Yeast and a few other microorganisms use alcoholic fermentation to form molecules called ethyl alcohol and carbon dioxide gas as a waste. The equation for alcoholic fermentation after glycolysis is as such. Pyruvic acid plus NADH yield alcohol plus carbon dioxide plus NAD+. The alcohol is ethyl alcohol. Lactic acid fermentation is the other type of fermentation. In many cells, pyruvic acid that accumulates as a result of glycolysis can be converted to lactic acid. This type of fermentation is called lactic acid fermentation. It regenerates NAD+ so that glycolysis can continue. The equation for lactic acid fermentation after glycolysis is as follows. Pyruvic acid plus NADH yields lactic acid plus NAD+. The first part of the equation is glycolysis. In lactic acid fermentation, pyruvic acid gets converted to lactic acid. So each pyruvate gets converted to a lactic acid molecule. You have two NADH molecules that were produced here that are going to come in here to help allow that conversion to take place. In doing so, you regenerate two NAD+ molecules. As long as you regenerate two NAD+ molecules, these molecules can come back into glycolysis to pick up electrons to form NADH. And ultimately, this allows you to continue to form ATP. This process is similar to alcoholic fermentation, with the regeneration of NAD+ doing the same thing, except remember, in alcoholic fermentation, we are producing two ethyl alcohol molecules plus carbon dioxide gas. That's it for today, biology. Have a great one.